and translation. Hiya. This video is all about transcription and translation. So we're going to start off with the big picture. So if this is our cell and this is our nucleus, first of all, in the nucleus is the DNA. Now I'm going to draw your DNA as one double helix. We know that in actual fact it's more than one piece. It's actually 46 chromosomes, but we simplify it as this one double helix here. So this is your DNA. Now your DNA is what we call your genome. Okay, and your genome has a few features that you need to know. So the first thing about your genome that you need to know is that it's universal. Now what this actually means is that it's the same in all organisms. So whether you're a fish, a bacterium, a person, a horse, whatever, it's the same. So that code is the same in all organisms. Okay. So what I mean by that is, you know how 3 bases code for one amino acid? So it would always be the same 3 bases that code for any amino acid. The second thing about your genome is that it's non-overlapping. So what I mean by that is we know that it's a triplet code. So let's say a section of it reads AAT, AAG, CCC, TTT. Okay, so you know that you would read that as triplet. So you'd read AAT, then AAG, then CCC, then TTT. So it's not overlapping in any way. If it was overlapping, you would read it slightly differently. So you would read the first 3 letters, AAT, the same as we just did. But then rather than then going on to AAG, you would reread starting from the second. So you'd say ATA. So the next 3 would be ATA. And then you'd start with the third one, TAA. And then you'd start with the fourth one. So 1, 2, 3, 4, AAG. And you get a lot more triplets out of it, but it's not like that. It's not overlapping, okay? It's non-overlapping. So this bit is not how it is. It's non-overlapping, which means each base is only read once. You never go back and reread a base. Each base is only ever read once. Non-overlapping. The third feature of it is that it's something called degenerate. Now, degenerate means that there's actually more than one triplet for each amino acid. So a triplet of bases, 3 bases, is the instructions for inserting one particular amino acid. Now we've actually got 20 different amino acids, but we've got 64 different triplets. And another word for those triplets, which we'll come on to later, is codons. So it's not like we've got 20 different triplets and 20 amino acids. We've got 20 amino acids and 64 triplets. So there has to be more than one triplet for each amino acid. Okay, so more than one triplet for each amino acid. Okay, so 3 properties there of the genome. Now, we've talked about this DNA being really, really large. In fact, it's so large that it can't fit out of the little holes in the nuclear envelope. So the nuclear envelope actually has little tiny holes called nuclear pores. And we need the genetic information to come out of the nucleus in order to get to the ribosomes. Now, you remember that the ribosomes, some are free in the cytoplasm, but a lot of the ribosomes are found on what we call the rough endoplasmic reticulum. So these are the little ribosomes, these little dots on the rough ER. Okay, so it's rough because it's covered in ribosomes. Okay, now we need this genetic information to come out to here. but there's no way that we're going to bring out the whole of the DNA, the whole of your genome. So instead what we do is we just make a copy of a small section of your DNA. In fact, a small copy of just one gene. And we call that molecule mRNA. And the M in mRNA stands for messenger. So it's messenger RNA. And that copy is just of one gene. Now, one gene is the instructions for coding for one protein. And we only need to make one protein at a time. So we only need to copy one little section of DNA at a time, one gene at a time. And then that little tiny piece of genetic material, that mRNA, is small enough to get out through the nuclear pore and to come out here and join onto a ribosome in order to make the protein. Now, the process of making that mRNA is called transcription. And the process of using this mRNA to make a protein is called translation. And they're the two processes that we need to look at, transcription and translation. So if we just go onto a new board, we're going to look at this overview of starting off with DNA, using that as a template to make a small molecule called mRNA, and using that as our instructions to make a protein. And the two processes that belong on those arrows are transcription and translation. Now, the processes happen, transcription in the nucleus and translation on a ribosome, which will be in the cytoplasm and maybe on the rough ER. Okay, so we're going to go through those two processes in turn. So let's start off with the process of transcription. So we're going to start off with our DNA, which is made of lots of genes. So we could split it up into sections and say each section there is one gene. So that could be a gene, which is the instructions for making one protein. And that's another gene, and that's another gene, and that's another gene. So we only need to copy one of those genes. So the first thing that we need to do is we need to undo this double helix. Now, we don't need to undo the double helix the whole length of the DNA, only just a little part of it like this. Now, when we untwist the double helix, that gets rid of the spiral. And then what we need to do is we need to break the hydrogen bonds. So you might remember that this double helix is held almost like a twisted ladder because the bases in the middle are held with hydrogen bonds. So we need to break the hydrogen bonds that are holding those two strands together. And another name for that is to unzip. What it means is to break the hydrogen bonds that are holding those bases together in that ladder shape. Now we need to know the enzymes that do this. Now the enzyme that untwists it is a type of gyrase enzyme, and the ASE tells me that it's an enzyme, and the enzyme that unzips it is a helicase enzyme, and again we've got this ASE which tells us that it's an enzyme. So enzyme names are really, really important. So this molecule of DNA here is still a double helix at the top here, but here we've got little bases sticking out that are no longer joined. Okay, we've unzipped it. That means we've broken the hydrogen bonds that would have held those bases together, and now they're exposed. Now, DNA is made of 2 strands. Now, the strand that we're going to use as a template is called the template strand. The other strand is called the coding strand. We're not actually going to use the coding strand, but that's what the other strand is called. So the next thing that happens is we need to start to make our mRNA. Now, this black molecule is DNA, and mRNA, as the name suggests, is RNA. So there are differences between DNA and RNA. Now, the way that we're going to make RNA, just the same as DNA is formed, is we make it out of things called nucleotides. Now, an RNA nucleotide is very similar to a DNA nucleotide. It's made of a phosphate, same, sugar, and a base. Now, the phosphate in DNA in RNA is exactly the same. It's this little circle here. The sugar in RNA is called ribose, whereas DNA it was called deoxyribose. And the bases can be A, U, C, or G. And you'll remember that in DNA it was A, T, C, and G. So this base here could be an A, or we could have a nucleotide where it was a U, or one where it's C, or one that was the G. So there's 4 different types. So this molecule is called a nucleotide. Okay, and specifically, this one is an RNA nucleotide. Now, if we were to draw them out like that every time, it would take us forever. So just look at the general shape. It goes down and across. So we tend to simplify them going down and across and draw them a little bit like L shapes. So remember that our template strand on our DNA now has these bases sticking out that aren't joined to anything. So what's going to happen is the complementary bases are going to attach on. You can see they're like the little L shapes. So we get complementary base pairing joining onto these little exposed bases on here. So what's happening is that RNA nucleotides bind onto the DNA bases. Okay. And then what they need to do is they need to join together. So here's our original DNA bases sticking out. And then we had our little RNA bases like this, and they're going to join together so that we end up with a chain, a polynucleotide strand. Poly, many, nucleotide, polynucleotide strand, just there. And that is our mRNA, so then the mRNA can detach, and this is our mRNA molecule that we've made. Now, the enzyme responsible for this is called RNA polymerase. Now, we're making a polymer. A polymer is a long chain molecule made of monomers. So the monomers that we're joining together were these RNA nucleotides, and we've joined lots of them together in a chain. So that's now a polymer. So it's polymer and then A's. Remember, enzymes end in A's. So our enzyme is called RNA polymerase. Now, when we're making DNA, it'll be made by DNA polymerase. Because we're making RNA, it's RNA polymerase. Okay, and we've used our template strand of our DNA as a template, and we've added the complementary nucleotides onto here, and we've joined them together, and then the mRNA is released. Okay, what will happen to your DNA then is it will wind back up again, and it's not damaged, it's ready to be used again. This mRNA that we've made is a small molecule, much smaller, as you can see, much smaller than the original DNA, and therefore it can fit out through the nuclear pores. So it's going to go out of the nuclear pores and is going to go and find a ribosome, probably on the rough endoplasmic reticulum, possibly free in the cytoplasm. So that's transcription. We've used a DNA template. Remember, this was DNA that we started with, and we've made a molecule of mRNA. So off our mRNA goes out through the nuclear pores off to the ribosome, where it's going to undergo a process called translation. So translation is going to use the mRNA as a template to make a protein. So a ribosome looks a little bit like this. It looks a little bit like a snowman because it's got a small subunit, just like the head, and a large subunit, which is like the body. Okay, and this is our ribosome. You might remember that eukaryotes have 80S ribosomes and prokaryotes have 70S ribosomes. So the first thing that's going to happen is that our mRNA molecule that we've just made is going to bind on to the ribosome. So here's our single-stranded small molecule of mRNA. Now, in the cytoplasm, we have lots of molecules that look a little bit like three-leaf clovers. And these molecules are called tRNAs. And the T stands for transfer. And what they're going to transfer is amino acids. Now, there's actually 20 different types of tRNA because there's a different tRNA for each amino acid, and there's 20 amino acids. Okay, so sticking onto this end here, attached onto the top end, is an amino acid. Remember, there's 20 types of amino acid, so there's 20 types of these tRNAs. Now, this is a molecule of RNA, which really is going to look like this, made of these RNA nucleotides with the little bases sticking out. However, it's been folded up. So rather than being in a straight chain like that, it's actually been folded up into this kind of clover shape. Now, most of those bases will be sticking inwards, and we don't tend to draw them, but they would be sticking inwards like that. But significantly, 3 bases stick out the bottom like this. Now, depending on what those 3 bases sticking out the bottom are, that determines which amino acid it can carry. So, for example, the first amino acid in any polypeptide chain, which is the chain of amino acids that folds to make a protein, the first amino acid is always an amino acid called methionine. Now, methionine is coded for on the mRNA by a triplet AUG. I always remember it because it's like August, AUG. Okay, so what we would need to get methionine is a tRNA that's got a complementary triplet. So complementary to A is usually T in DNA, but this is RNA, so it'll be U. U is the equivalent of T, so that's complementary would be A, and G's complementary pair is always C. So the first tRNA that we're going to need every single time is always going to be the one that's got UAC on the bottom. So what we've got is our ribosome looking like this. We've got our mRNA attached here. I'm going to read these bases that are sticking out in groups of 3. Now, 3 bases on an mRNA is called a codon when it's on an mRNA. 3 bases on the mRNA is called a codon. And what we need is the complementary anticodon. The 3 bases that stick out from a tRNA are called an anticodon. So what we need is, as we said, the codon will always start with AUG. So what we need is this tRNA here with UAC there, UAC, to bind on because AUG and UAC are complementary, and it will bring with it a certain amino acid. Okay. I'm going to get a new whiteboard because it starts to get a bit messy. Let's make sure we can draw it nice and clearly. So now what we've got is our ribosome with our mRNA here. Remember that 3 bases on an mRNA are called codons. Then we've got our tRNA coming in up here. carrying a specific amino acid. And whatever 3 bases we've got here, we need a complementary 3 bases up here. So that codon will bind there because we've got an anticodon on the tRNA that's complementary to the codon on the mRNA. Now, the next amino acids, all the other amino acids are just, they are a specific sequence, but they're different for each protein. So let's say we've got, onto the next 3, CCC. So just by trial and error, different tRNAs will try and bind with that. And eventually, we'll find one that's complementary, that's got the GGG as its anticodon, that will bind, and will bring another amino acid with it. Now, those 2 amino acids at the top there will join together, and the bonds that form between amino acids are called peptide bonds. Okay. Now, this first tRNA is finished with is going to go off to go and fetch another amino acid to transfer another amino acid. So it's not going to be needed anymore. So in our next diagram, here's our ribosome. What I'm going to do is I'm going to show that the ribosome is moving along the mRNA. So you can see that the mRNA was just here, and now the mRNA is sticking out the end because the ribosome has moved along. So we've got our sequence of bases along here. So the next one's GGG. Okay. Now that first tRNA that delivered that first amino acid has gone. It's floated off into the cytoplasm without its amino acid. It's going to fetch another amino acid. The second tRNA will still be bound just here. I'm not going to draw the detail of everything, but draw that one in there. And then another tRNA is going to come in and bind here. So with its complementary anticodon to the codon, bringing another amino acid. Now the amino acid from this first one is still going to be attached here. So we've got amino acid number 1, 2, and now we've added 3. So this was 1, this was 2. Yeah, so 1 is there without the tRNA is gone. 2 is still attached, 3 is now coming in. And we get a peptide bond to join number 3 on. And you can see that we've got this growing polypeptide chain. Now eventually, what's going to happen? Oops, sorry. Eventually what's going to happen is that we'll get right up to the other end. The tRNA, the ribosome will have moved all the way along the ribosome right to the end, and we'll just be putting in our last amino acid. Now we're going to end up with a whole chain of amino acids going back this way. Okay, because we had from the triplet code, we put an amino acid in from this codon, from this codon, from this codon, this codon, this codon, this codon, this codon, this codon. So each one of those codons will have coded for a specific amino acid being added to the chain. Now eventually, this will disconnect here. Okay, this tRNA will float off. Go transfer another amino acid for another protein that's being made somewhere. And this here is our finished chain. Now, a chain of amino acids is called a polypeptide. And you'll remember that polypeptides fold up into proteins using the primary, secondary, tertiary, and sometimes quaternary levels of protein folding. Okay, so this finished polypeptide here now needs to fold up into its specific 3D shape, and then it will be a finished protein. Okay, and that's transcription and translation in its kind of simplest form. Now I'm just going to talk you through one more thing. Now there's one more step that's sometimes mentioned and sometimes not. So you remember that we went from DNA to mRNA. Now, sometimes we might say that we go to something called pre-mRNA, then we go to something called mature mRNA, and then we go to protein. So this process here was transcription, which we've talked about. And this process over here was translation, which we've talked about. This process here is called splicing. And that's what I'm going to just add in now as an extra level of detail. So basically, the mRNA that we've made here contains some sections that do code for amino acids to make a protein, but it also contains some sections that don't code for protein. And the two types of sections that it's made of are called introns and exons. Now, the coding parts are the exons. These are what we call the coding regions, the bits that code for the sequence of amino acid. And an intron is what we would call a non-coding region. So if we start off with our pre-mRNA, our pre-mRNA is this chain of RNA nucleotides, like this. We could actually separate that into areas that are exons that will code for protein, and some areas that are introns that won't code for anything. The next bit might be an exon. Now, in the process of splicing, what we need to do is we need to remove the introns and then stick the exons together. So simply what happens is the exons just come together like this. So we've got one exon and another exon that just get joined together. The intron is removed completely, and the enzyme that does that is called an endonuclease. Okay. Shush, I'm just making a video. 2 secs. Okay, so that process there just fits in between transcription and translation. Okay, and that's